I'm curious. I am curious. Curious.
By nature.
Welcome to Curious by Nature. I'm Jessica Johnson from Newswise. When you want to edit someone's DNA, you need a set of molecular scissors. That's where CRISPR comes in. The highly specialized technique can find a specific point on the genome, snip it in two, and splice in a string of new code. Now researchers are looking into how this method can be applied to the health of our microbiome and what the good bacteria in our bodies can do to keep us healthy. It's all about engineering the microbiome to better understand how it affects our well-being. The use of CRISPR and the microbiome are what we will talk about today. Dr. Ronda, welcome. Please introduce yourself.
I'm Carlotta Ronda, and I'm a principal investigator at the Innovative Genomics Institute at UC Berkeley, IGI for short. You will hear it a lot, but I will say that IGI, it stands for Innovative Genomics Institute at UC Berkeley.
Tell me about your work in general.
I lead the research lab at IGI, where my team is investigating how the bacteria, they live in our body. The most notable ones are the microbiome, the gut microbiomes. Everybody has heard of them. And so how these bacteria interact with our body and our physiology, how they affect our health and disease. And then eventually, once we know the mechanisms that these bacteria elicit in our body and triggers either disease or they help us to be healthy, we wanted to basically harness this knowledge to develop novel microbiome-based therapies.
Could you tell me more about that recent study in Nature Protocols about bacterial genome engineering using CRISPR?
Yeah, so I don't know if everyone is familiar about CRISPR. Maybe people have heard about it, at least the acronym has reached the mass media, but CRISPR is actually originally was a bacterial immunosystem that scientists actually one of the major contributors in discovering this CRISPR system and harness the power of this system is the head and director of the Innovative Genomics Institute, Jennifer Doudna, who actually was awarded the Nobel Prize in 2020 with Emmanuel Champetier about this technology. So they studied this like immunosystem in bacteria, and then they discovered that it's a powerful system that can be used to edit genomes. Because how it works, it's a molecular scissor of the DNA. So basically, it's the ability to target a specific molecular scissor to cut the DNA at a specific site into the genome of every organism. At this point, the technology was unveiled, or like the repurpose of this natural system as a technology was unveiled in the, I think the seminal paper was 2012. And so since then has been shown that works in pretty much every organism. And now actually very recently, the first gene therapy for sickle cell anemia was approved thanks to actually the effort of Jennifer and the IGI. And so now we can actually edit genes in human to treat disease using this kind of molecular scissor that cut the DNA and allow to repair it.
Can you tell me more about your recent research study in Nature Protocols, and that was about the bacterial genome engineering using CRISPR?
That's a new avenue to target microbiome. So we need to start asking why are we doing this? Like, why are we using CRISPR? Why we are editing the microbiome? Because, people might wonder, what's the purpose of editing microbiome? We do have that, so we don't need that. But the reality is that substantial evidences have shown that the microbiome plays a crucial role in our physiology. It's not just there to help us to digest the food. It seems that is involved in the onset of many diseases, including diseases that you would never consider being linked to the gut, like neurodegenerative diseases or neurodevelopmental disorder. More and more data are coming out showing the key contribution of the gut microbiome and the metabolites and the molecules that they produce, and their effect into different, in basically systemically in every organ of our body. So for me, has been very almost like mind-blowing and very game-changing, realizing that bacteria in our gut can change our brain chemistry and then affect our mood and behavior. And for me, it's really fascinating. However, the problem that we have right now is that we don't know how, why, like how do they do that? So like there are a lot of correlational data. Like we see a specific composition of bacteria in the microbiome that is associated or linked to a disease or to a mood disorder or neurodegenerative disease, but we don't know how, like why. And that's why we need to edit the microbiome. We need to engineer the microbiome because we need to understand what it's doing, and we need to understand this mechanism. One of the real reason why CRISPR systems or CRISPR technology has been so revolutionary in science and research. But the really disruptive power of this technology in research has been the ability to ask fundamental questions of how our genes function, how we function, or how plants' genes function. Because for example, in the early 2000s, there was the Human Genome Project has started, where we decided to sequence the entire human genome. Sequencing technology was at the early stages, but we moved a long way from since then. But so we decided to the scientific community decided to sequence the human genome, and they were convinced that by knowing the sequence of our genome, we would be able to understand disease and health. We were able to understand our physiology. The reason, but the fact is that we can't. That's just a code. It's just like it's basically if you didn't know a language, and you were given a book in that language, and you don't understand what those words means, and that's what basically is. So if we don't know what genes are doing in the context of our physiology, then the code is useless.
Is that new that someone has used CRISPR on the microbiome?
CRISPR, since at early stages, because it comes from bacteria, people have used it in bacteria, and then they extend it into human cells and insect and plants. However, in the context of bacteria, has been used in what we call monocultures. Monocultures are just like one type of bacterium that grows in the lab. And it's completely taken out of context, and it's just like looking at a problem, not in the right environment. It's like if you were taking out a cell from your organ or your tissue and study that single cell without understanding the context in that tissue. So CRISPR was not yet applied to microbiomes or to microbial communities because they're very complex, because microbial communities are communities. So they're not all the same cells. There are different species of bacteria that they live together, and every species has its own genome. So like the genetic diversity, it's outstanding. And also one of the major issue, for example, of microbiome or like a gut bacteria growing in our gut, is that it's very difficult to isolate them and cultivate them in the lab, because they grow anaerobically. So that means without oxygen. And so it's very difficult to have them to grow as like in a lab conditions. You need specific equipment to be able to grow some of these these organisms that they don't tolerate oxygen. And even in that scenario, is very difficult to still cultivate a lot of them, because they require nutrients that we don't know yet they are requiring those, that they're present in their native in their native environment, like the gut. And so it's very difficult to understand what is the best media or food that to give to this bacteria to grow.
Basically, your research was looking at microbiome, uh, a specific type of bacteria that was in the gut. And you were able then to use CRISPR to then change a bacteria.
Few years ago, I have developed a platform that allow us to directly modify bacteria in the gut, directly in what is called in situ, in their native environment. And this, to our knowledge, was the first demonstration that of these type of technologies, that the first demonstrations of the ability to engineer or edit bacteria directly in the gut without isolating them and try to engineer them, which we know isolating these bacteria is very challenging. After developing that platform, I wanted to increase the precision at which we were targeting these bacteria and we were engineering these bacteria. So I combined this platform that it's called MAGIC. That stands for metagenomic alteration of gut microbiome by in situ conjugation. It's a crazy, long, difficult words, the explanations, but in a nutshell, we basically harness the ability of bacteria to exchange DNA with one another. And so we use it, we engineer it to deliver a specific new gene that they codify for a new function directly in situ in the gut. We did this experiment in mice. So we actually give these mice our what we call donor. It's kind of like we can consider that as almost like a probiotics. It's not a probiotics, but if we wanted to just like imagine what it would be, it's kind of a bacterial cell that is innocuous. It's not harmful, and it's just transient. It would just like pass through. And so this cell is the donor of the DNA and the new function that you are going to engineer into the native microbiome.
You had multiple research studies that had different steps. Can you explain those?
Yeah, so the first study is a study where we develop a platform that allow us to deliver new functions directly in the native microbiota in the gut, in situ. Following that study, we were able to develop a technology harnessing CRISPR, the ability of being like these reprogrammable systems, to be more targeted, to basically have these what we call base pair resolutions in the way we edit these microbes. In this second study, we use a very specific CRISPRs that is called Cas or CRISPR-associated transposons. And the difference between the CRISPR that I explained earlier, where it's a molecular scissor that cuts the DNA, and then we can replace it with the healthy gene, this CRISPR system is slightly different because bacteria, they're not able to repair DNA when it's cut. They're not really good at, like us. We're very good at repair the DNA when it's cut, but bacteria, they're not. So when you use the original CRISPR or the OG CRISPR, which is called CRISPR-Cas9, in these in bacteria, it's very toxic. So it will kill the cell eventually because they cannot repair their DNA unless you introduce another molecule of DNA to be able to repair it better. However, it's like multiple step procedure that it's not user-friendly for editing bacteria. While these new cast systems are slightly different. They're still reprogrammable. So you still have one RNA sequence that is guiding the CRISPR at the specific site that is identical or is like complementary with the sequence of the RNA. However, instead of cutting the DNA, it take a piece of DNA that is next to it, that it comes with it, and then introduce that piece of DNA into the genome. So instead of creating a cut that is difficult to repair, it opens the DNA and introduce already the DNA. So it's not toxic to bacteria. Okay. But it's still very targeted and use the same concept of these RNAs or RNA-guided reprogrammability, where we have an RNA that is complementary to the target sequence, in the genome of the bacteria, but introduce a gene instead of cutting. So we first developed a technology that could deliver this gene, because they could deliver new functions, which could be this gene. So we delivered these genes. And then now we developed this technology to be able to introduce genes into the genome without killing the bacteria. And combine this together, we created a novel editing system that this is able to introduce new functions into the native microbiome in a very ultra precise way. And not only is able to introduce new functions, because you can use this reprogrammable enzyme, and they're called reprogrammable transposase discussed, to introduce these new functions. We can also disrupt the genes when we introduce this DNA sequence, because you can imagine that, yes, you can target a specific sequence in the DNA and then introduce the functions of interest. But wherever you introduce that function, that DNA sequence will be open. And then if there is a gene like if the sequence is inside a gene, you will break the gene, because you will introduce another piece of DNA that, will completely disrupt the coding sequence of that gene. And so we'll knock out that gene. So using this technology, we can directly introduce and remove genes and at the base pair precisions in the mammalian gut, in the native site.
So, and then the third phase.
And the third phase. So now that we have this technology that is able to have this ultra precise in vivo microbiome editing, we develop a platform that is translational. We use these systems to target pathogens during an active infection. So imagine you have these active infections, and the pathogens or bacterial pathogens are pathogenic because they have genes that codify for toxins. And that's why they make us sick. So the idea is to use our delivery method and combined with our precision method to basically remove the toxic gene from the pathogens during the infection, so you don't get sick. You'll be fine. So even though you have an active infections, you won't feel any symptoms. Your body will be fine. And at the same time, we introduce a new functions. And these new functions is the functions that prevent the pathogen to stick around and help the immune system to fight the pathogen and remember the pathogen for future infections. So using this system, what we call backtrains, we are able to basically intervene during an infections. This was tested in mice, where mice were not getting sick because we removed the genes that were making them sick. And then also using this system, we introduced the functions that allowed that made these pathogens kind of like helpers for the immune system.
So basically, I mean, hypothetically speaking, still, this is a possible therapy like an antibiotic or a vaccine, but it could even be when someone is sick, and it doesn't have to be taken prior to getting sick. It can be taken when someone has the symptoms, and it can basically change that whole bacteria so they don't feel the sickness anymore.
Yeah. Correct. But also will help to protect for future infections. So you don't get sick. If eventually you get reinfected, then you will be immune.
What are some of the sicknesses or some of the, I guess, bacteria that are the most common or most deadly that are found or originate in the microbiome?
For example, Escherichia coli or E. coli infections are still a significant global health concern, contributing to approximately 950,000 deaths annually. And for specifically enterotoxigenic E. coli, which is EC or ETEC, it's actually one of the leading cause of diarrhea in developing country and among travelers. Travelers' diarrhea, that's one of that. Or if you get infected, you won't be able to get sick. And so you can still enjoy your vacation.
Fascinating. Thank you so much, Dr. Ronda.
Curious by Nature is a production of Newswise. More than 7,000 journalists get the Newswise wires. Visit newswise.com and find out how to spread your news today.